Hello and welcome to BFRB Awareness Week and Teaching Tuesday with our presentation today of brain mapping and BFRBs. I'm Jen Monteleone, Interim Executive Director, the TLC Foundation for Body-Focused Repetitive Behaviors. Some quick housekeeping items before we get started. This is an educational webinar offered in a safe and welcoming environment for everyone attending today. Questions for our guest should be asked through the Q&A feature, and your questions will be answered at the end of the webinar. If you have any tech challenges during the webinar, please use the chat box to message one of our TLC staff members, and we would be happy to help you. Our guest today is Dr. Joseph O'Neill. Dr. O'Neill is a professor of child psychiatry at the UCLA Semel Institute for Neuroscience. His research entails magnetic resonance, especially MRS, investigations of the brain in pediatric and adult psychiatric disorders and their treatment, with an emphasis on OCD and substance abuse. Please help me welcome Dr. O'Neill. Hello.
Hello, thank you for that gracious introduction.
You're so welcome. I'm going to go off camera now, and I'll be back to help you out with Q&A.
All right, so if you can all see the PowerPoint there. Okay, great. And we'll get started. Today we're going to talk about brain mapping, neuroimaging, and BFRBs. And we're going to discuss some results from research that was very generously funded by the TLC Foundation, with a little bit of help from NIH and some help from the Red Cap project at the University of Chicago. Just a quick note, there's been other BFRB neuroimaging research before and outside the TLC, many great contributions, some of them done by our own BPMI investigators and collaborators. They have informed and inspired us, but we will scarcely talk about them. Today's focus is TLC research. Here's our outline. We'll discuss the TLC pilot study in pediatric hair pulling disorder, HPD, and then we'll go on to results from adults who have both either HPD or SPD, skin picking disorder from the big BPMI study. And then we may go on to look at some of the more recent pediatric results on hair pulling disorder and skin picking disorder from the BPMI, but I doubt we'll have time. And then we'll summarize and then a bit of Q&A at the end. Let's get started. This is the first paper from the TLC pediatric hair pulling disorder pilot. It's a paper on electroencephalography. It was the second ever paper of any EEG paper of any kind on hair pulling disorder. And so a little bit of background on EEG. Electroencephalography, EEG, is brain electrical activity recorded at the scalp. You know that inside your head, in your brain, the neurons are firing all the time, changing the electric field inside your brain constantly. A small fraction of that electrical activity seeps out to the scalp, where we can pick it up with electrodes, like this gentleman wearing an electro cap with 64 electrodes. And from each electrode, you can record how the electrical voltage changes over time. Here's the scale down here, 140 microvolts per 2 seconds. And sometimes it goes faster, high frequency EEG, sometimes it goes slower, low frequency EEG. And we can record the EEG during various cognitive tasks. Like the gentleman here is watching visual stimuli being projected on the screen, and he's responding to them by pressing on the key press pad. We can see how all of that affects the EEG. The main way of analyzing the EEG is to break it up into its frequency components, the fast and the slow components, like gamma waves, for example, very fast, beta, fast, but not the fastest. Then there's the famous alpha waves you probably heard about. When you're relaxed, you have a lot of alpha EEG on your scalp. And then you can calculate how much electric power is inside each of these frequency bands, as we call them, for a certain interval of time at a certain electrode. And the amount of power in each band can vary with different cognitive functions that your brain executes. For example, the theta power down here, one of the slow waves, has been linked to inhibitory processes, inhibitory control. Inhibitory control is very important for BFRBs. If you feel an urge to pull or pick, for example, do you have the power to resist that urge? Do you have enough inhibitory control? So we wanted to know, what is theta power like in people with BFRBs, in this case, hair pulling disorder, and children with hair pulling disorder, children in adolescence. And this is what we found. It's kind of a pretty sophisticated way of displaying the results, but I'll show you. We looked at electrodes at different parts of the brain that are related to motor action and sensation. For example, electrodes that are on the scalp above the supplementary motor area in the cortex, electrodes above the right motor cortex, primary motor cortex, and then others that are above the superior parietal cortex. And for each of these, we looked at the different frequency bands here on the vertical axis, and then on the horizontal axis, we looked at time. So how do the frequency bands vary with time? We measured how much power was in each band, and you can see that's plotted based on the color bar over here. Bright red colors for strong positive electrical power, and then deep blue colors for strong negative electrical power. We were interested in the theta band of EEG, which is the solid black box here. And we looked at this in people, typical people without hair pulling disorder. We looked at it in kids with OCD, obsessive compulsive disorder. And we looked at it in people with TTM, trichotillomania, which is the old Greek name for hair pulling disorder, HPD. And if you look at these inside the box, you can see there's different amounts of red. You've got about half of it is red for the healthy controls, so good amount of theta power, and but even more for people who have hair pulling disorder, and hardly any for the kids with obsessive compulsive disorder. We are including these kids, by the way, because, as you probably know, hair pulling disorder, skin picking disorder are somewhat like obsessive compulsive disorder. They're not the same thing, but they're kind of like it. They're thought of as obsessive compulsive spectrum disorders, sort of a family resemblance, but not identical. And we looked at these powers and the other electrodes too. And our findings were that we had some elevated theta power in these brain motor centers in HPD. And I didn't show the plots, but someone also correlated with the HPD symptom severity. For example, elevated power in the supplementary motor area, SMA, that's located here in the brain. If you're looking right down the middle of the brain and off on this kind of what we call the mesial side of it, the middle side of it. This is the supplementary motor area. It's close to the very top of your brain. That's involved in initiation of voluntary movement. That had higher power, theta power, which perhaps corresponds with more difficulties in motor inhibition. And or it did correspond with more severe HPD symptoms. And then we had more power over the right motor cortex, the primary motor cortex that's involved with execution of movement. There it was theta power stronger in HPD than controls. And over this is where the motor cortex is located, this M1 here in the pink. And then the superior parietal cortex is not in the middle of the brain, it's out on the lateral surface, the side surface of the brain. Here we're showing it in orange, it's towards the back, and in orange, the superior parietal cortex, that involves sensory input from the hand, very relevant to HPD because you pull your hair with your hand, and then you feel how tightly you're pulling and so on. That's sensory input coming back into your hand. Theta power there was higher in hair pulling disorder than in controls, and the higher theta power corresponded to more severe symptoms. So that was kind of interesting. Now, there's another way of analyzing EEG besides breaking it up into the big rhythmic waves. You can also look at short-term changes in the EEG record. For example, if I flash a light in your eye, there'll be a series of little blips that occur over a second or so in your EEG trace. And these things are called evoked potentials. They're unleashed by sensory stimuli or motor actions or so on. We were interested in an evoked potential that's called the error-related negativity, the ERN. And this is an evoked potential that rises up when you realize that you made a mistake, that you committed an error. For example, if I was showing you on a video screen, I was broadcasting capital letter A there. Every 2 seconds, I'd show you A, and then once in a while a B instead of an A. And your job was to press a button every time you saw the B. You'd be watching, you see A, then B, okay, press, B, okay, press, and A, and press. Oops, drat, I made a mistake. I pressed on an A. If you did that, there would be a big negative electrical wave that would rise up over the frontal scalp, the scalp in the front of your head, and that's called the error-related negativity. It peaks at about 70 or 100 milliseconds after you made your response. Now, very interestingly, if you look at, here's a plot of the error-related negativity at one electrode. Here's the voltage versus time, and you can see around here, around the 70 milliseconds, so this big peak comes up. The interesting thing is this was studied in people with OCD versus controls, typical people without OCD. And the ERN in the OCD people is like twice as large as in the control people. And this is one of the most dramatic findings in psychiatric neuroimaging here, actually. It's done by a guy named Bill Gehring from Michigan, who was a visiting professor at UCLA a few years ago. So the ERN amplitude, the size of it, is high when you realize you committed an error. And in OCD, the ERN amplitude is very high, and that's because people with OCD are hyper-conscientious. They feel responsible for everything, and it's a huge disaster for them when they commit an error. So that kind of makes sense. That's been seen in adult and pediatric OCD. So over here, this will be the only HPD finding, BFRB finding that's not TLC-sponsored research. Roberts et al. 2017, that was done, I think, at University of Philadelphia. Marty Franklin was one of the co-authors on this, and as you know, he's been a tremendous contributor to BFRB clinic and research for decades now. Anyway, they decided to look at the ERN in HPD, and they did it in adults. And what they found was, in contrast to an OCD, where it's larger in HPD, hair pulling disorder, the ERN is actually smaller. This is a plot of it here, and it's smaller. So, I mean, that may be because, fortunately, people with hair pulling disorder are usually not hyper conscientious. They don't go crazy about it, making small mistakes and that sort of thing. So in adult HPD, ERN amplitude is below normal. Very interesting. We asked, what about pediatric HPD? So we did a study of that in a small sample of kids with, again, TTM, hair pulling disorder here, and some OCD kids, and then some typical neither OCD nor HPD children and adolescents. And here's the plot of the ERN at one electrode. Notice we're plotting it, the other people were plotting it upside down with the negative up. We're actually plotting it the proper way. The y-axis is negative down. And when we do that, we got for the OCD kids, we get a big, huge error-related negativity, just like we've seen in the adults. And for the kids with HPD, though, it's not smaller. It's bigger than it is for the healthy controls in the blue here, but it's not nearly as large as it was in the adults. And if you look at here, these brain maps, here we're recording the error-related negativity at many different electrodes on the scalp, and then we're kind of interpolating between them to create a map out of it. And the OCD kids have this big blue wave in the front of the scalp. It's the electrical field, electrical voltage at the time in this square here from 0 to 100 milliseconds after committing the error. They've got a great big ERN wave here, and then it's quiet in the back of the scalp. The control subjects have almost nothing in the front and some big positive wave in the back. And the HPD kids are kind of an intermediate form between OCD and control. They got a little bit of an ERN out here, and then a little bit of kind of an orange in the back, similar to this red in the back, the positive wave that you'd see in the healthy controls. So they're kind of intermediate, which is interesting. Okay, so that was a big workup for somewhat modest finding, but there's a point to that. This is typical of research. It's a good example of how science works in actual practice. We scientists tend to overgeneralize. For example, we say, maybe the ERN is the same in all OC spectrum disorders, maybe the same in OCD as it is in HPD. But nature is always splitting and subdividing. It tells us, no, the ERN is different in HPD than it is in OCD. So we try another round. We say, maybe the ERN is the same in pediatric as in adult HPD. Nature subdivides on us again when we get the result and tells us, no, the ERN is different in pediatric and adult HPD. This is one reason research takes so long, because we move forward in these little incremental steps, like, you know, okay, HPD versus OCD, okay, adult versus child, and so on. And that's very typical of research. The second paper that came out of the TLC pilot, however, it gave us a little bit more than we expected. This is a paper about magnetic resonance spectroscopy, MRS. And this one was not the second, this was the first, as far as we know, ever HPD paper on spectroscopy. And magnetic resonance spectroscopy is a neuroimaging method that measures the amounts of neural metabolites in each brain region. Here on the left, we've got an MRI. You've all seen MRIs. This is a little section, a so-called axial section, like a horizontal section through the brain, like this. And we've got a grid, this green grid superimposed on it, which is made up of voxels. Voxels are volume elements. They're similar to like the pixels on your computer screen. Pixel is a picture element. A voxel is a volume element. So it's like a pixel, like a little square, except it has some depth to it. So it's like a little cube. In this case, the cubes are about 0.5 cc's in size. And inside of each of those little voxel cubes, we acquire what's called the magnetic resonance spectrum on the right. That's a series of peaks here. We're plotting the magnetic resonance intensity as a function of the frequency that the scanner operates at. And at each different frequency, we have a different peak that represents a different biochemical, a different neurometabolite, as we call them. And so the frequency it's at tells you which biochemical it is, and then the size of this peak tells you how much of it is there. So it's a way of measuring the concentrations in vivo inside the brain. The two neurometabolites we were interested in were glutamate, or GLU, GLU, and gamma aminobutyric acid, GABA. We've got their chemical structures here. Glutamate is the main excitatory neurotransmitter in the brain. It's also a major neuroenergetic metabolite, so very important for brain energy. And it happens, however, to be a main source of excitotoxicity. If you get too much excitatory activity in your brain, it kills off your brain cells. So glutamate is your friend, but it's also dangerous. GABA is the main, fortunately, we've also got GABA. It's the main inhibitory neurotransmitter in the brain, so it can calm the glutamate down. We asked, what are glutamate and GABA levels like in the brain in pediatric hair pulling disorder? Could high glutamate be related to stronger urges to pull, for example? Would low GABA be related to weaker resistance to pulling? I mean, it's kind of simple-minded hypotheses, but something you can look at. And we're also interested in whether glutamate and GABA were changed by behavioral therapy, BT, treatment for hair pulling disorder, or even if the levels of these metabolites could predict response to BT. Here's some of the brain areas that we looked at, that we acquired the magnetic resonance spectroscopy from. This is another, again, an axial section through about the middle of the brain. Up here is the front by your forehead. Back here is the back of your head. And then we've got the left and the right. We tend to reverse left and right in some of these images here. And one of them is called the pregenual anterior cingulate cortex, PACC here. That has to do with positive affect and reward, things that you like and you enjoy. And the others here, we've got the caudate nucleus here, CD, that's this little thing in here. These two, they look like crab legs. If you notice, the front segment of the crab leg here is the caudate nucleus, and the back segment looks sort of like a wind sac here. That is called the putamen. Next to it is something called the globus pallidus. And then down here, this little egg-shaped thing is the thalamus. These are all subcortical nuclei. And they're involved in things like action selection, behavior switching, and habit learning and unlearning. Action selection, like you want to eat a banana, which banana do you peel off the bunch? Behavior switching, you're heading down the freeway, you've been pressing the gas for a long time, hazard comes up, okay, ease off the gas, ease off the brake, switching behaviors. Habit learning and unlearning, very important for BFRBs. Let's face it, BFRBs in common sense terms are bad habits, very ingrained habits. And in therapy, you try to unlearn those habits or learn other wholesome habits to cover them over. Okay, so here's some of the results that we got. In the putamen, this nucleus here that we said looks like a wind sac. Well, first of all, before we get to that, the result of the behavioral therapy. We've got here each diamond here, red diamond is one of the patients. It was a small number, 10 patients. And we've got their scores on this particular severity measure for hair pulling disorder before and after. You can see everybody went down at least a little bit, and 9 of 10 of them were classified as responders, which we defined somewhat leniently, a greater than or equal to 25% decline in this scale. So this therapy that was developed by and implemented by Dr. Paris was effective, at least in this little group that we had there. But interestingly, at the same time, we had this drop in severity. There was this big rise in the putamen in the levels of GABA in there, which I thought was quite interesting. In the pre-treatment here, this is trick pre-treatment, that's hair pulling disorder before treatment. Levels were about the same as the controls, but they went up after the treatment. So it's almost like they had to go up to above normal levels in order to reduce the hair pulling behaviors. Well, the putamen is active in motor suppression of behaviors and in habit learning. So you ask, maybe does more GABA and putamen improve motor suppression or improve bad habit breaking or replacement habit learning? Maybe it does. These are all just speculations. But we also found that at baseline, that in the pregenual anterior cingulate cortex, the PACC, that's here, front of the brain, that baseline severity was higher for higher levels of glutamate in that structure and in that cortex, and lower for lower levels of GABA. So maybe more inhibition there was better, and less excitation there was worse. And a similar correlation for glutamate, at least, was seen in the thalamus. Now, the pregeneral anterior cingulate cortex, the PACC, is involved in positive affect, reward, pleasure. So maybe does the PACC excitation increase? Does excitation of it increase what one might call the rewarding effects of pulling, and therefore aggravate HPD severity? And rewarding effects of pulling, what I mean there is maybe after you pull out a hair, when you pull it out, it's painful, but then there's a relief that comes right after. Or also that the pulling is like a relief of boredom, release of stress. This can also be thought of as a kind of rewarding effect. We also found in the caudate nucleus up here that glutamate pre-treatments and the GABA actually predicted the response after the treatment. So high pre-behavioral therapy GABA predicted a poor response, and high pre-behavioral therapy glutamate predicted a good response. Now, the caudate is known as the gear shift of the brain. It helps you to shift from one action to another. So again, a speculation, does high caudate glutamate mean a less sticky brain, one that's better able to respond to therapy? Perhaps, but it's interesting that it could predict the response, at least in this little group. So we took all of these findings and we integrated them into a sort of model. All of these subcortical and cortical structures that we just talked about, they're interconnected in these loops that are called CSTC, cortical striatal-thalamocortical circuits. And they excite each other with glutamate and they inhibit each other with GABA. And we made it kind of a model out of that. We made one for the pre-treatment state and one for the post-treatment state. Okay, so that was kind of, that was fun. Let's go on now to the BPMI, the big study, adult findings under the big study. The first paper from that is this was an MRI paper, which is currently under review. It's not quite published. We'll see. Probably will get published soon. And quickly, let's look at our BPMI database. How many subjects we were able to recruit. This is the largest neuroimaging study of BFRBs that's ever been done by anybody in the world, as far as I know. And there was 183 BFRB people, 82 with hair pulling disorder, 56 with skin picking disorder, and 45 who had comorbid hair picking and skin picking disorder, and then 56 controls without hair picking or skin picking disorder. And so we acquired MRIs from all of these people, and we fed them into a marvelous program that's called Free Surfer up here, FS. And we took all these hundreds of MRIs and dumped them into the program. And of course, that meant we had to wait in the queue on the supercomputer, wait over hours and days. And then because it was a big demand on computing resources. And once the computer finally started, then it crunched the numbers. That goes on for hours and days. Then usually it crashes a few times. And you have to go and debug it and rerun and debug. And again, this is why this research takes forever, because you're constantly having to mess around with this stuff. But when you get the results, you get something like this. This is this MRI here, with gray matter, white matter, subcortical gray matter, CSF. The FreeSurfer program parcelates it automatically into all the individual cortical and subcortical regions, all the different gyri and lobules and that kind of thing. And you can see them color-coded here, 72 different cortical regions, and then in the gray, 16 different subcortical regions. And for each cortical region, it calculates what's the average thickness, what's the average surface area of that region, and what's the volume of that region. And then it calculates what's the volume of the white matter that's underneath that. And it also calculates for the subcortical nuclei, it just calculates what's their volume. So it's a lot of data, although we're not even done at that point, because we have to QC all of these individual subjects for all these regions. And they usually do look startlingly close to what you would identify with the naked eye, but they frequently also have errors and so on that we have to correct and throw them back in and so on. Okay, anyway, so from looking at all that, those data, we got some findings, some effects involving the cortical thickness and subcortical volumes for HPD and SPD people versus healthy controls. And they're plotted here. And the top, we've got, again, these axial MRI sections. And this is kind of an average MRI across all the subjects. And down below, we've got, again, average MRIs. We have them for TTM. Again, this is hair pulling disorder, SPD, skin picking disorder, and the people who had both. Down here, these are coronal sections of the MRI. It's like sections parallel to your face going back like this. And on the axial and the coronal sections, we are plotting here. It's like a statistical parameter, the F statistic. It's basically measuring how different is the thickness or of the cortex or the volume of the subcortical nucleus in this group relative to the controls. And we found here, you can see in the red means it's a very big difference, and deep blue means almost no difference. And we've got some nice red for the caudate and the putamen for our hair pulling disorder people. And we've got some nice red for our SPD people in the, in the right IFG, that's the right inferior frontal gyrus. Those are the main findings. Just to show you again where these regions are located. This is the famous Gray's anatomy here, 160-year-old series, but it still has some of the best sketches for the brain and other organs. These are the frontal lobes of the brain up here, and this is the lower part of the frontal lobes. It's called the inferior frontal gyrus or the inferior frontal cortex, divided up into three sections: pars orbitalis, pars triangularis, pars opercularis here. The left inferior frontal cortex famously is involved in speech. If you get a stroke there, you can get an aphasia, a loss of speech. The right inferior frontal cortex has a function of motor response inhibition, pulling you back from something that you were about to do. You're about to press a button. Oh, no, I shouldn't press that button, pull back. This makes perfect sense, that because when we have right now I'm doing a monologue, but if we're having a dialogue, I talk for a while, you listen, then you talk, I listen, I hold my tongue. So we're constantly using the tongue, holding the tongue, and the two, the left and the right part of the brain, are working in tandem to help us conduct the conversation. Here's the same results we were just looking at, just plotted in bar graphs since it might be clearer. Looking at the volume in the HPD people versus the controls. We have these higher volumes in the green here for the controls of the left and the right caudate and the left and the right putamen, and the lower volumes in the red for the people with TTM, which is HPD, hair pulling disorder. And then over here, looking at the right inferior frontal gyrus, again, we've got higher volumes in the green for the controls, and then lower volumes in the orange here for the people with skin picking disorder, and all three of the subsections of the inferior frontal gyrus, you've got the lower values of the thinner cortex there. Okay, so we saw effects of HPD and SPD on the MRI volumes and cortical thickness in the caudate, nucleus, and the putamen. Left and right caudate volume, HPD was less than controls. And perhaps that means that we said the caudate is the brain's gear shift. Perhaps that means a dysfunctional caudate might mean that people with HPD could get stuck in a rut, maybe kind of on a hair pulling binge type behavior, that sort of a thing. Left and right putamen, the volume again was lower in HPD than controls. Putamen is active in suppression of motor behaviors and in habit learning. Perhaps people with HPD have difficulty suppressing pulling, difficulty unlearning the habit of pulling. And then in the right inferior frontal cortex, we had thinner cortex in all three sections for SPD, then for controls. Well, the right inferior frontal cortex is active in motor response inhibition. Maybe people with SPD have difficulty resisting picking, perhaps for that reason. Okay, so that was the first MRI paper on the adults. Here's another paper on the adults from the BPMI study. This is from the clinical data. We identified some clinical subtypes. Remember, I said nature is constantly subdividing. Even when we have a disease, a disorder identified, we get all these subtypes, which are important for precision medicine, personalized medicine. And we found, by using advanced statistical analysis of the BPMI clinical data, we identified some subtypes of hair pulling disorder. These are kind of interesting. Sensory sensitive subtype with highly focused pulling, but infrequent and low intensity urge to pull. Although awareness subtype, automatic pulling and pulling to emotional triggers. That was the most common one. And then an impulsive perfectionistic subtype, pulling to control unpleasant feelings and feeling unable to resist the urge to pull. We had a couple of SPD subtypes as well. There was an emotional reward subtype with strong, frequent urges to pick, auto picking plus picking to negative emotions, and little sense of control. Then there was a so-called functional subtype with mild SPD, weaker urges to pick, and little distress. And the typical controls, people who neither pull nor pick, they're no fun, but we had to include them anyway. So we wanted to see if the MRI was consistent. MRI findings were consistent with any of these subtypes, and we did find a little bit. We again dumped all the data into the all the MRIs into the FreeSurfer program, and then analyzed them vis-a-vis the different subtypes. And we found, FreeSurfer found a few effects of subtypes on volumes of brain structures, and left lateral occipital cortex volume, low awareness subtype, greater than sensory sensitive subtype and controls. Lateral occipital cortex is not just active in vision, but also in touch and proprioception, meaning your sense of knowing where your limbs are in space and how much your joints are bent and that kind of a thing. Right lingual cortex, there was a finding in impulsive perfectionistic subtype, lower volume than sensory sensitive subtype or controls. Lingual cortex is involved in a number of functions, including response, inhibition for negative emotions, and reward processing. Maybe that has something to do with HPD. And then some small effects for the SPD subtype. So there are pictures for this too, but they're not really that impressive. So we'll just leave them out and move on. Okay, then we come to two fMRI papers that came out of the BPMI initiative. The first one involved associative learning. First, a little background on what functional magnetic resonance imaging, fMRI, is. It turns out that if you have oxygenated blood, like in your arteries, the MRI signal is a little bit stronger for oxygenated blood, like you see in this red curve here, than it is for deoxygenated blood, like in your veins. You know that, well, you know how that works. You have the heart, the lungs fill your blood with oxygen, and then it gets pumped out into the body, and then the body eats up the oxygen, and you have deoxygenated blood that comes back in the veins to the heart, and so on, right? But the fMRI signal is a little bit stronger for the oxygenated than for the deoxygenated, and that's a slight effect, but we're able to pick it up with our fMRI scans and do so routinely. Now, the interesting thing is that the brain regulates its own blood perfusion. So if part of the brain is more active, some activity you're doing, you're reading, you're running, you're playing music, you're doing this or that, whatever part of the brain is responsible for that activity gets flooded with more oxygenated blood at the expense of the other parts. And because it gets flooded with more oxygenated blood, you get a little bit stronger fMRI signal out of those parts that are currently involved in that particular activity, which means that if we have you conduct an activity and conduct an fMRI scan during that activity, we can figure out what parts of your brain are responsible for performing that activity. And that's the basic idea behind fMRI. In our study, we did a very complicated task. It was called an associative learning task. We ran a number of these trials, hundreds of different trials where we would present something to you on the screen, the computer, on the projection screen, and you have to push a button in response. And in each trial, we would pick one of these 6 abstract geometric objects. Like, for example, in yellow here, this thing looks like an Aztec headdress, for example. And we'd project it to you, and you would have these 4 buttons on a keypad or your 4 fingers, and you would press, and maybe you press a 1, guessing, oh, maybe 1 is for the Aztec headdress. And no, it turns red. That's the wrong one. And we show you more symbols and more symbols. The headdress comes back up again. You press 3. Bing. Oh, green. Great. 3 corresponds to the headdress. I learned that one. Super. But then after you've learned it, we switch it around. And then it comes up again. You press 3. And now it's red. Oh, no, they switched it. And you have to go and learn again, pressing each time it comes up. Bing. Okay, 2. Now it's been switched to 2. So that's the task, associative learning task that involves this initial learning and then reversal learning, reversing what you did. Now, people with OCD, we make lots of references to OCD because it's the same kind of family of disorders, hate that when you switch things on them with the reversal learning. So we were wondering what is that like in BFRB? Do BFRB brains act differently during this task than people who don't have BFRBs? And the answer we found out is yes. If you look over here on the right, we had to combine the HPD and the SPD, like merge together the 2 groups to get an effect in this case versus the controls. And we're looking at the fMRI signal now here for the initial learning minus what it was during the reversal learning condition. And we find out here in the light gray what the signal is for the healthy controls. It was a lot stronger in the controls than it was in the BFRB people in 3 main regions. One was the caudate nucleus here, CD. One was the middle frontal cortex and several subsections of the middle frontal cortex here. And one in pregeneral anterior cingulate gyrus, which we mentioned, and its neighbor, the anterior middle cingulate gyrus. Here they are mapped out. Here's where the caudate nucleus is. This little orange blob is where we saw the difference, a significant difference between BFRB and controls. And then we have two blobs here. One is for the pre-genital anterior cingulate cortex, and then one for its neighbor a little bit behind the anterior middle cingulate cortex. And then finally, the middle frontal cortex, you can see it here on this axial section higher up in the brain. Over here and over here is where that's located. Here's another shot of where it's located. It's the brain seen from the side. It's in the middle of the frontal lobe here. It's responsible for what we call executive functions, working memory, cognitive flexibility, inhibition, abstract reasoning, things like that. Flexibility, inhibition, probably important for BFRBs. The two parts of the cingulate that we mentioned, we were saying before, the PACC is involved in processing the positive affect, happiness, pleasure. The AMCC back here, that's kind of a fear center of the brain, also a pain center. And between the two of them, between pleasurable things that attract you and fearful and painful things that repel you, these two kind of work out your approach avoidance behavior, whereby it's modulated by your willpower. You can still approach something that you're afraid of. You can still stay away from something that you enjoy if you put your mind to it. And we would love to understand how these mechanics work in BFRBs. If we could understand that, maybe we'd have the therapy down. Okay, so we saw weaker bold contrast, the smaller difference between initial learning and reversal learning in HPD and SPD than in controls. And the caudate and putamen, maybe that has to do with difficulty in habit formation during initial learning in HPD and SPD. We saw it in the middle frontal cortex. Maybe that has to do with organization, updating, and set shifting being more challenging for people with HPD and SPD. We saw it in the PACC and the AMCC. Maybe initial learning was less pleasant and required more effort in HPD and SPD. And finally, there was one more paper we'll go having to do with reward processing. Maybe I'll go straight to the results here. This is basically a task, an fMRI task where we're running low on time, so I'm moving ahead, where you are expecting that you're going to win money, like $1 or $5. And what does the brain look like when you're expecting you're going to win money on this? And we found out there's a stronger fMRI signal in BFRB people than in controls when you're expecting to win money, anticipating winning in the left and right inferior frontal cortex. And when you're expecting to lose money, there's also a stronger signal in the left and the right inferior frontal cortex, but in a couple other regions too, the right fusiform gyrus and the right inferior parietal lobule. This is where the fusiform gyrus is located. It has to do with stimulus recognition within category identification. This is where the right inferior parietal lobule is. It has to do with interpreting tactile information. And so again, during anticipation of win, stronger signal in BFRB than the controls in the inferior frontal cortex. Inferior frontal cortex is active in stop-start behavior, reward, and punishment. Maybe these functions operate differently in BFRB than controls. During loss, we had the same effect in the right inferior frontal cortex and these other two regions. So maybe these functions like stimulus recognition, interpreting, tactile info, and so on are maybe different in BFRB than controls. Okay, we'll do our overall summary and then we'll get on to the questions. Overall summary of all that we've looked at in this little lecture today. Neuroimaging of BFRB has been highly under-researched, but the TLC-sponsored investigations have now made great strides towards addressing that need, with many further results forthcoming. I've got lots of coming attraction slides and so on that I could show you if we had time. Most results, now But importantly, for every effect that I've shown you, there's a ton of effects where the HPD people were just no different at all from the typical control. So most results to date in HPD and SPD are normal, as expected. Doing these studies, one, we want everything to be normal for your sake. And but number two, if there is something that's not typical, we want to find out what it is, in case it can help us diagnosis, prognosis, treatment, and so on. We expected most of it to be normal because although BFRBs can be severely impairing in your daily life, at the same time, you don't have paralysis, you don't have sensory loss, you don't have visual defects, you don't have auditory defects, you don't have tremor, you don't have seizures, all of the, you don't have hallucinations, all of these things that you can see with brain disorders or atypical development. So we're looking for very specialized things. It's like a needle in a haystack kind of a problem. Having said that, interestingly, positive findings keep popping up in the same brain regions, caudate putamen, PACC, and so on. And these are regions with functions that are intuitively related to the clinical phenomena of BFRBs, motor inhibition, habit learning, and so on. In terms of specific findings, we've done the first EEG study of pediatric HPD. We found effects involving theta power and involving the ERN in relevant motor and sensory motor brain regions. We've done the first MRS study of HPD, found that behavioral therapy was effective, at least in a small sample. We saw that increase in GABA after behavioral therapy. We saw that glutamate and GABA varied with HPD severity. Caudate glutamate could predict response. We developed the first brain-based model of HPD in this treatment. We, with the THC and the BPMI, TLC and the BPMI, have done the largest ever MRI study of HPD and SPD. We found several effects there involving volumes and thicknesses of cortices, and we identified intuitive clinical subtypes of HPD and SPD. We've done the largest fMRI study ever of HPD and SPD, and we found fMRI effects in these relevant regions, suggesting how ideas of how cognition in BFRB may be different than in typically operating brains. And then we didn't look at them, but there's several emerging findings in the smaller pediatric HPD, SPD sample involving smaller white volumes in the inferior parietal cortex and these other effects. So we've got lots of findings to contemplate in BFRB week. And with that, I'm ready for questions, Jen, if we have any.
We have a couple. Let me get to them. I think that you might have answered a little bit of this in your presentation, Dr. O'Neill, but we do have a couple questions related to GABA supplements and glutamate supplements being helpful in treating BFRBs. Are you able to share a little bit more about that?
I'm not. I mean, I haven't. The short answer is I don't know. There's so many complex biochemical reactions that take place that sometimes it's simple-minded to think that, well, I'll just take more GABA and I'll take more glutamate. And because first of all, these things get digested in your gut, and then they have to make it up into your brain, and then they undergo all kinds of complex reactions. So a priori, we can't know for sure whether these types of supplements are going to work, but they might. It's an empirical question. Really, you'd have to do a clinical trial just of the supplements. And I don't know if those have been done and what the results are. Certainly something worth doing. And I'm sure people are running out and doing it regardless of what the science says to date.
Okay. We have one more question. Can you explain a little bit more about the learning piece of the findings?
Okay, the associative learning task,
I'm
the,
not quite sure. It just says learning piece.
the learning
So we might
piece. Okay, let's
extrapolate it to
see.
the associated learning. Let me see if we can get a little
Okay.
bit more detail from the person who asked that question.
All right, maybe I can bring that, I can bring those slides up more again. Let's see if we can. All right, let's see. We can go back to that. We can look at the task again, if that's what you mean. Here we go. Yeah. Okay. This is the associative learning task here. So on each trial, well, I can explain the task again if people didn't get that.
Dr. O'Neill, that is what was asked. Yes, that's the piece. If you could cover
Okay,
it,
good.
please, that would be great.
Okay, well,
Thank
here,
you.
good. I didn't read this text on the bottom. Then I can read it now. That may explain everything. So in each trial, one of 6 abstract figures is projected on screen. You're looking at it, you're lying in the MRI scanner, and there's a screen that you can see above you. And one of them gets selected on each trial, like this case, this pointy one here, in yellow is selected. And then you've got 4 keys on a keypad, one under each of your 4 fingers on your right hand. And the figure gets projected up there on the screen, and you have to kind of guess, does the one go, which number goes with, which key goes with this figure? Is it the 1? Is it the 2? Is it the 3? Is it the 4? We don't tell you in the beginning. We show these, we keep showing you these images, these figures, and you have to keep pressing and guessing, right? Until you figure out which it is. And whenever you guess wrong, it turns red. And when you guess correctly, it turns green. And then after it's been shown to you however many times, 5, 10, 15 times, then you figure, oh, okay, the Aztec headdress is number 3. And just when after you've got it down, you think everything's great, then we switch it on you and change it to a different number. And we don't tell you that we're doing that. So you have to try again, pressing the numbers until you get them figured out. If you got the game mastered for all 6 of these, you'll have each one of them, what's their number. Some of them can have, since there's 6 here and only 4 keypads, you can have 2 figures with the same number. Maybe one of these others is also 3.
Dr. O'Neill, the question for more clarification is, once you go through this abstract learning task, at the end of it, how does it relate to what we're trying to determine with BFRBs? How does it correlate? What does it mean?
Well, let's see, I think we kind of got this. The idea is to see how quickly you can pick up a pattern and then how quickly you can rearrange it afterwards. So in one way, it's testing your rigidity. How fixed are you? Like if you learn it once, are you able to switch over to another pattern, right? And there can be a kind of rigidity in BFRB, and that you get the same behaviors over and over again repetitively. So it's a way of measuring your behavioral flexibility, ability to shift out of one behavioral scheme into another. So that's part of it. And the regions that showed the effects of BFRBs had to do, as you see here, with regions that execute certain functions that are kind of relevant to BFRBs, like habit formation and set shifting and so on. Or how effortful is it for you to learn the initial pattern? Let's see, is that making any sense?
That's great. And actually, we're a little over time, so it's time for us to start wrapping up. So I'm gonna join you again, and we can unshare the slides if you would, please.
Okay.
Thank
We'll stop
you.
share. There
Thank
we
you
go.
so much. That was an absolutely fascinating presentation. Thank you so much, Dr. O'Neill, for giving of your time and your expertise to help us better understand how our brains are connected to our behaviors and the research that TLC has been funding to help us learn more. We really appreciate your time today. So thank you so much for being here.
It's been my absolute pleasure.
Thank you to the amazing TLC staff for putting this event together today for the community, and to all of you for being here today. Thank you so very much. Don't forget to help us by completing the post-event survey, which is now linked in the chat. Remember that completing a post-event survey enters you into the drawing for a BFRB Brave box valued at over $150. There are still more BFRB Awareness Week events this week, my friends. There's something for everyone. You can learn more on TLC's event page. The link is now in the chat. And it's not too late to wear your BFRB pride. So going over to our BFRB merch page through Bonfire and take a look and see if there's something for you. 10% of every sale is donated back to TLC to support programs like this. And that link is now in the chat. And then finally, if you enjoyed attending today, please consider supporting TLC by making a donation. This helps us continue to offer programming like BFRB Awareness Week and more. You can make a donation now at bfrb.org/donate. Thank you all again for joining us. We look forward to seeing you soon. Have a great evening, everyone. Bye-bye.